Какой микрофон будет работать? Оба работают, да? Good morning, dear students. Good morning, dear professors. We're glad to see you online, to see you offline. Today, we would like to present you professors from Chinese University, Guangliang and Gao Zhichen. And today we'll speak about pharmacology for students who already studied this subject and for those who is just going to study it. So, dear professors, you are welcome.
So, hello, can you hear me?
Yes.
Yes.
Okay. Yeah, I'm Guang Liang. Today we invite Professor Gao Zhichen in our group in Wenzhou, and give you interest of this topic, this speaking, and he will take his research here. Okay, let's welcome Professor Gao Zhi, Professor Chen.
Hello. Hi. Can you hear me?
Yes, sir.
Yes,
sir.
Good morning.
Okay. Yeah. Okay. Thank you for the introduction by Guangliang, and this is the greatest honor for me to have a speak here. So let me share my screen first. Okay, so okay, let me start with my talk. So today I'm going to see something about the fibroblast growth factor. It's a very brief introduction about these factors. So why is FGF, which is a fibroblast growth factor? Because my research is measuring the FGF. So today I will going to see something about FGF for all of you, and at the beginning, and for the rest, I will see some research project by myself in the recent years. So for the first thing is the discovery of the FGFs. From the PowerPoint, you can see at the beginning it's 1974, but actually at the beginning it's 1940. Somebody found very interesting things that they get some liquid from the brain and put it into a cultured fibroblast cell lines, and they found that the cell line were proliferation very, very quickly. So, but at that time, nobody knows what things doing those proliferation works there until to the 1974. The Danish, they found these factors, they can directly stimulate the fibroblast cells to grow very fast. So that's why this growth factors is named as fibroblast growth factors. But even from the 1974 to 1986, all the FGFs we get is from the bovine brain. Almost 600 bovines can only get 500 microgram FGFs. That is very, very little, and the protein is very, very expensive at that time. Until 1986, the scientists from the US, they first identified the DNA sequence of BFGF, which is now is called FGF2. So after that, the people did one thing is that to do the recombine the FGF protein from the E. coli sister. So based on this, all the pharmacy scientists, they are trying to express the FGF protein outside and get the huge most amount of the protein and to discover the drugs. So until to 1996, the group from China, which is my supervisor, Xiaokun Li, they did a major work on the expression on the discovery of the drugs of FGF. So they get the first approved drugs of FGF, which is called recombined bovine FGF2. It's used for the wood herring. So until this time, the first FGF is going to the clinical application. So next is called that FGF. Why is so important? Because first of all, the FGF proteins is a very important classes of proteins which the evolutionary conserved, even from the nematode to the human. All of those animals, they all have 4 class of FGFs. Even from now is known as FGF, they have 23 members. So all of them have 23 members in there, and their sequence is the similar is very similar to each other. So that's why this protein is thought that it's very, very important for the peoples in to do the research and to use it in the clinic. So actually, this is said that FGF have a wide range of clinical application value, like in the heart, in the brain, in the eye, in the ear, in the nose, in the mouth, in the bone, and even in the kidney, in the lung, in the liver. So all of those tissues, the Fgf families, they plays a very, very important role in their development, in their the proliferation, endocrinology and metabolism, regulation, and so on. So this makes FGF as a very, very hot topic in all over the world to for the scientists to do the research there. So then we go to another slide. So this slide is called that the drug development of FGFs by our team, which is our team means the team from my supervisor, Xiaokun Li professor. He is majored in the drug discovery of FGF families for almost 30 years. So the first drugs, like I introduced before, is recombinant bovine, a basic FGF, which is the one they show here, the RBFGF2. So this is the first thing we funded and get approved at 1998. But at that time, these drugs also have some program to use, because the major program is immunogenicity. So in order to improve the immunogenicity program, the second step, we are going to do a recombinant human basic FGF, which the resource is a human. So this protein is totally solved the problem of the immunogenicity program. And this drugs is get approved at 2002. But this drugs also have some problem, because these drugs you can see here is basic FGF. So as you know, the wood environment is always contain an acid environment. So the basic FGF always not very stable in the wood environment. So in order to improve the stability of the drugs and make it can apply to more and more wood conditions. So we just keep to improve it, and we make the recombinant human acid FGF and get improved at 2006. So this is the drug development story of the whole FGFs. And now the following, I will go into a brief introduction for the FGFs. So the first one is classification of FGF. As I mentioned before, FGF totally have 23 members at the beginning. So the first one we found is FGF2, because at that time we found it is basic FGF, and then we found acid FGF, and because A is in the front of the B, so at that time when we found when the people found FGF3, so they named A FGF as a FGF1 and then FGF2, and in the following until the 2001, they found the lasted FGF members, which is called FGF 23. So at that time, totally it goes to 23 members for FGF. But actually now this FGF family, all the family is divided into 3 different subfamily. One is called paracrine FGF, one is called endocrine FGF, and another one is called intercrine FGF. But now the people think that this intercrine FGFs, FGF 11 from FGF 11 to FGF 14 is not exactly FGF families, because their acting is different to the traditional FGFs. All those FGF, I will say, I will mention it later. All of those paracrine and endocrine FGFs, they work as they need to bind to the receptor, and then they can stimulate the kinase activity of the Fgf receptor, and then they were going to get the activity. But for the endocrine FGFs, they are different. They do not need to bind to the receptor. They just, and even they do not secrete outside of the cells. So it interaction into the cells. So now people always think that FGF 11 to FGF 14 is not exactly the FGF subfamilies. So I put it far away from the major FGF subfamilies. Now it's called FGF homology factors. So for the main FGF subfamily, the paracrine and endocrine. And the paracrine subfamily, they also divided into 5 different subfamilies, which is called FGF1 subfamily, FGF7, FGF4, FGF8, FGF9. All of those subfamily belong to paracrine FGF, and for the rest, FGF 19, 21, and 23, which is my major work, is belong to the endocrine FGF. So what's the difference between paracrine FGF and endocrine FGF? It's because of the heparin binding site, heparin binding affinity. Because the paracrine FGFs, we can see from the structure. Today's talk, I will talk a lot about the structure, because my major is structural biology. So if we take a look of all the FGF families members of the structure, you can see from the figure, this blue means the heparin binding site of the FGF. We make it in blue. You can see from FGF1 to FGF 22, actually 18. So they all have a lot of blue in there. So they all have a lot of heparin binding site. So they can bind to the heparin very strong. But if you can see here, the endocrine subfamily of FGF, you can see FGF 19, 21, and 23. All the FGF heparin binding can bind to the amino acid which can bind to the heparin is very, very little. So the FGF 19, 21, 23, which is a classic endocrine FGFs, they works very, very bad affinity to the heparin. So you may ask, what is heparin? Why it's the heparin binding site makes it a paracrine and endocrine? So I will talk about this next. So first, you can see a figure here. This is a SPR result for the paracrine FGF2 and the endocrine FGF 19. This is 19, sorry, 19, 21, and 23. You can see this experiment is to measure the binding affinity between two different molecules. So we code the heparin into a chip, and we just throw away the FGF. Then you can see if they have a stronger binding, you will see a very strong signal here. And you can see the FGF2, you see a very stronger signal, right? But for the endocrine FGFs, FGF 19, 21, and 23, their heparin binding site, their heparin binding affinity is very, very low. So this thing makes the two different subfamily, which paracrine, endocrine, FGF, did a totally different works in our body. So we take a example like paracrine. You can see a figure in the right. You can see the paracrine FGF8. They were, because of the stronger heparin binding affinity, and the heparin is a sugar that major in the between the cells and the cells. So when you express the cells express the FGF, paracrine FGF, and they scream the paracrine FGF outside of the cell lines, the cell will the heparin between the cells will catch the FGF immediately, and then all those FGFs cannot go far away from the cells which express them or scream them. So they can only act to themselves or to a cell line that next to it. So that's why it's called paracrine and the paracrine FGF. So for the endocrine, you can see the FGF21 here. The cells express and secrete it outside the cells. Because of the heparin, it cannot, because of they do not bind to the heparin very stronger. So the heparin cannot catch all of the FGF 21 or the endocrine FGF immediately near the cell line which screen it. So all the endocrine FGF can secrete into the bloodstream. So from and it can follow all our blood system, go to everywhere, all of our body to act. So that's why it's always like FGF. Take an example, the FGF21. It is expressed in the liver, but it can act at adipose tissue. That's why the FGF subfamily, FGF families is divided into paracrine and endocrine. So now we know is all about the FGF factors, right? The fibroblast growth factors. So how the FGFs works? Like I told you before, the FGF they must bind to a receptor, which is called Fgf receptors. So Fgf receptors is belong to a RTK subfamily. What is RTK? RTK is receptor tyrosine kinase. So we can see here is a receptor tyrosine kinase, the superfamily. So this superfamily totally contains 58 proteins and 20 families in human. We can see our friends, the Fgf receptors is belong to here. So there's three different three different domains in the Fgf receptors. The first domain is, you can see here, is the cell membrane. So there's one domain is that extracellular domain, which is outside this cell membrane. And they contains 3 Ig-like domains outside the cell members. And they have a very short transmembrane domain with a very short peptide, like maybe 30, yes, contain a 30 peptide, 30 amino acid with this transmembrane domain, and then they have a intercellular domain, which is also called the kinase domain. So the kinase domain, they can act to their downstream factors and phosphorylation the downstream the substrate, and then they can transfer the signals and stimulate their functions like proliferation or metabolism regulation. So how the Fgf interact with Fgf receptors, and how the Fgf receptors get activated and transfer the signal. This is the major question, the major project for the structural biology to start. So that's what my major research. So, but at the beginning, I will see something about before funding. So the Fgf receptor, the major thing is that their extracellular domain. This extracellular domain is used to bind to the Fgfs, and then the Fgfs will make this domain dimer, and then maybe the kinase domain can close to each other, then they can phosphorylation each other, and the signal will come and go to the downstreams. So we can see here the Fgf receptor, the extracellular domain, they have 3 different domains, the D1, D2, D3 domain. The D2, D3 domain is most important domain for Fgf to bind, and they contains a D2, D3 and D2, D3 linker. So the D1 domain, which is useless for the cell for the signal to transduction, but it is a very important domain that can inhibit the ligand binding. So when the Fgfs is not in an active condition, the D2 domain will go down and bind to the D1 domain will close to the D2 domain and block the Fgf to bind to the D2, D3 domain. So totally the Fgf receptor family, they have 4 different genes, which is called Fgf receptor 1, Fgf receptor 2, Fgf receptor 3, and Fgf receptor 4. But the Fgf 1, 2, 3, the Fgf receptor 1, 2, 3, they have another alternative spliced regions and make it into totally 7 isoforms, which is B and C isoform. So only receptor 1, 2, 3 have these 2 different isoforms. So Fgf receptor families, they totally have 7 different isoforms. And then we can see here, this 2 is a dimer structure for the paracrine Fgfs. Sorry, I make a long here. So this in the right is my supervisor in the New York University, who is Mohammed Musa. He is solved the crystal structure of FGF2 and FGF receptor 1C and heparin together forms a dimer here at 1999, which is published in Cells. You can see here the orange one is FGF2 and the green one is FGF receptor 1C. So you can see the FGF2 can bind to the D2. This the top one is D2 domain and the lower one is D3 domain. So FGF receptor can bind, FGF can bind to the D2 in the middle of D2 D3 domain and forms a binary complex, and then you can see the heparin binding. The heparin is in the middle of two binary complex. So when the heparin comes, the two binary complex will came together and forms a dimer like this, and the two different FGFs can close to each other and make the intercellular domain, the kinase, they close to each other, then phosphorylate each other. But at 2000, there is a scientist named Brown-Dales. They also published another dimer model. They also solve another crystal structures, which is FGF1, Fgf receptor 2, and heparin binding, and heparin sulfate. They published in Nature in 2000, and from their structure, one thing is the same, is that FGF is binding to in the middle of FGF receptor D2 and the D3 domain, right? But the major difference is that their heparin is in the middle of two FGFs, not in like our model that FGF heparin is in the middle of the receptor. Their heparin is in the middle of FGF in the ligand. So they forms a two receptor, two FGF, and one heparin model. Our model is two ligand, two receptor, and two heparin. So this is a major difference. And in the following almost 20 years, people almost accepted the dimer model of in the left, which is more symmetric. So at the first, we solved this canonical model, which is FGF bind to the FGF receptor, and heparin makes this binary complex forms a dimer, and this is called the paracram FGF. They bind to each other. So as we think that when the two receptors they close to each other, how the intercellular domain, the two kinases they act to each other, maybe act with the substrate will be their downstream substrate. So in the 2016, even my supervisor Xiaokun Li, they solved another structure, which is the kinase domain of the FGF. They bind to their substrate. The green one, this orange one is their substrate PLC gamma. So they find the FGF receptor 2 kinase domain with PLC gamma CSH2 domain, the interaction structures by the X-ray. So they find that two FGF receptor, when they close to each other, their intercellular domain, the kinase domain, they can close to each other and phosphorylate. After that, the phosphorylate FGFs, here you can see the tail here. This is the C tail. So the phosphorylate FGF is here. This tyrosine is by phosphorylated. So this phosphorylated residues can, like a finger or like a hand, they go outside and bind to their substrate. Like here, this substrate is called PLC gamma. They can catch the PLC gamma close to themselves, and then another FGF receptor, which when they form a dimer, they are very close to each other, right? So the another FGF receptor, they have a kinase domain, which is the kinase domain, the activity pocket here. Here is a pocket that he can phosphorylate the substrate. So you can see the PLC gamma, the C tail of the PLC gamma, which is the tyrosine, serine of the PLC gamma. Their C tail were put into the activity pocket of the kinase domain, and then biphosphorylated by the green FGF. So this is a model that how FGF they interact with their downstream signal. So to now we find that the total thing, the paracrine FGF, they can bind directly to FGF receptor, and the heparin make a hand inside, and they push two binary complex together to form a dimer and make two receptors close to each other. And then when the receptor close to each other, they can the kinase domain of the receptor can phosphorylate each other. And then after phosphorylate, the two receptors, they act like a friend. One is to catch the substrate like PLC gamma, and another one to phosphorylate it, and then the phosphorylated substrate is activated, and then they can get the signal. So this is the major ACT model for the paracrine FGF. So till now, all the structural research is based on the FGF, the paracrine FGF. But from the 2000, the endocrine FGF was founded, and there's still no any structure models, any structure resolved that can see how paracrine endocrine FGFs is work. Is they work similar to the paracrine or not? So nobody knows. Okay, so we take the as the following. We take the major endocrine FGF family members, FGF23 as a factor that to study the endocrine FGF, how they interacted with FGF receptor and activated FGF receptor. So in the following is my recent works. FGF23 is a like endocrine factors is a hormone that is screened by boron oxide, and they can screen to the blood system, go to the everywhere to take action. But the major action is a separation of the phosphate reabsorption and vitamin D homosynthesis in the kidney, and also inhibit the perasorodized hormone, the production in the parasoroid, and also can regulate the calcium and the sodium homeostasis. So this is a very important. This is a very important factors in the endocrine FGF families, but different to the paracrine FGFs. The paracrine FGF, I told it before. First, the endocrine FGF, maybe like the FGF23, they bind very weak to the heparin, right? So another way is that if you compare with paracrine FGFs, here in this with the same experiment we did before, but this time we code the FGF receptor in the chip. So we can see FGF2 and FGF23, we compare their binding affinity to the receptor 1C. You can see the KD, which is can indicate the binding affinity of two different molecules. The value is more lower, means the binding affinity is more high. So you can see the paracrine FGF2 to FGF receptor 1C, the KD value is 58. But if you go to the FGF23, you can see the KD value is 648, almost 10 times less. So those two means that FGF23 cannot can very hard to directly bind to the receptor and even to heparin. So he cannot exactly act as same as paragram and FGFs. So how did it works? So then people found another co-receptor for the FGF23, which is called alpha-closol. He is a very important factors for FGF23 that can offset its FGF23's poor heparin and FGF receptor binding affinities. You can see here, alpha-closol is a short transmembrane proteins. They have a very short transmembrane domain, and their major domain is outside, which is KL1 domain and the KL2 domain. So this type of alpha-closol, we call it alpha-closol transmembrane alpha-closol. So when did the alpha-closol found? At the 1997. At the beginning, they were found as a anti-aging and as a aging genes. So when you knock out the alpha-closol genes in the mice were age very quickly, and their lifespan is very short. So at that time, people named the alpha-closol as a fifth goddess of Greek mythology, which is called Closol. So this is where Closol name came from. But then you can see, if we coat the chip with FGF receptor 1C and alpha-closol together, we can see when we use the FGF23, the KD value is go to 27.4 nanomolar, which is almost 20 times than FGF receptor 1C alone, even is higher than the paragram FGF with their receptor. So it means the alpha-closol is the transmembrane domain of the alpha-closol is a co-receptors for the FGF23 to bind to the receptor and to transduction the signals. And the alpha-closol, which is expressed in the kidney distal tubes and parasolidize gut and the brain. So there is another kind of alpha-closol in our body, which is they will be cut by the ADAM proteinase. To cut where? Cut close to the transmembrane domain. Then after the cut, the alpha-closol will secrete to the blood system and go to everywhere. So this kind of alpha-closol, we call it alpha-closol actor domain. So to our research published before, everybody thinks that only alpha-closol transmembrane domain is act as a co-receptor for the FGF23. But the alpha-closol actor domain cannot act as a co-receptor for the FGF23, and they are act as a like I said before, as a glycosides and to act as a FGF23 independent activity to like aging and something else. But there's two major programs still exist and nobody can answer it. First, why deficiencies of alpha-closol or FGF23 give rise to a virtually identical phenotype? What does this mean? So people did the knockout. Sorry. People did a experiment, knockout mice. They knock out alpha-closol alone, knock out FGF23 alone, or knock out FGF23 and alpha-closol together. All the mice, the phenotype is the same, no difference. So it means FGF23, even FGF23 or FGF alpha or alpha chromosome, they do not have any, independent activity to each other. Their activity should be the same. This is the first thing. Another thing is that why FGF23 regulates the phosphate reabsorption in the kidney proximal tubes, but alpha chromosome, the transmembrane alpha chromosome, is expressed in the kidney distal tube, right? This is very strange. So we think that this alpha chromosome actor domain should be act the same as transmembrane alpha chromosome, which can be also as a co-receptor of FGF23. So we first express this actual domain of alpha chromosome, which we cut this transmembrane domain, transmembrane alpha chromosome, transmembrane domain, and we get this actual domain of alpha chromosome, and we did a Western blot study, a stimulation study with cell lines, which is here you can see the HEC293 cells, they only express the receptor without alpha chromosome, and we add the alpha chromosome active domain we add the protein we expressed, and in the right, this is HEC293 cells with which we transfected alpha chromosome, the transmembrane domain, the four alpha chromosome. We can see here, even the actual domain of alpha chromosome can stimulate when FGF23 is added into the system. So it means the alpha chromosome active domain can act as a same co-receptors for FGF23. Even in the in vivo study, we also see that the alpha chromosome active domain can induce the FGF23 signal. So this means soluble alpha chromosome can act as a co-receptor for FGF23. So that's why we in the following we use this soluble alpha chromosome, the active domain of alpha chromosome as our protein. Then we also express the FGF23 protein and FGF receptor 1C protein. We put those three proteins together and get a very pure FGF23, FGF receptor 1C, and alpha chromosome active domain, three protein, they bind to each other. We get a ternary complex, and we use this ternary complex to grow the structure crystal, and to use the X-ray to solve the crystal, solve the structure. Then we get this structure. So you can see here the crystal structure of FGF23, FGF receptor 1C, and alpha chromosome ternary complex. From here, you can see FGF23 is also bind to the FGF receptor in the middle of D2, D3 domain, which is totally similar to the paracrine FGFs. But one thing is different, is that the chromosome. So the chromosome is act like a co-receptor for the FGF23. So you can see here in the movie, the orange of the FGF23, you can see they have a long C-terminal tails. They bind to the middle of FGF alpha chromosome to KL1 and KL in the middle of KL1 and KL2 domain. They forms a very stronger interaction, and except that FGF alpha chromosome, they have a alpha 1 beta 1 strand here. They forms a receptor binding to with the receptor in the top in the bottom of the structures. We call this peptides, we call it receptor binding arm. So from this structure, we can see that why alpha chromosome is so important for FGF23 to bind to the receptor 1C, because alpha chromosome, they bind to the receptor 1C first, and then they also at the same time they bind to the FGF23. So when those two binding is formed, then FGF23 can bind to the receptor 1C. That's the structure what tell us. So in the next, we first to find that two alpha chromosome, the two different domain, KL1 domain and the KL2 domain. The KL1 domain in the structure, we can see the KL1 domain is sitting in the top of KL2 domain, and there are a lot of amino acids. They forms a hydrogen bond between each other to form a very stable interaction. And when we mutate those amino acid to the alanine, the alpha chromosome cannot form a so stable structure, and they will lose their activity. And next is a major thing, as I mentioned before. The people think that alpha chromosome can act as a glucosidase that to do FGF23 independent activity. Why? Because of if you can see here from the structure, you can see even KL1 domain and the KL2 domain, their structure is very similar to the glycosidase hydrolysis A clade, this family. What's the figure? It's like they have 8 beta strand in the middle of this structure, and 8 alpha helix outside the middle. It's like like a ball in there. So there's 2 different KL1 domain and KL2 domain is all like this. So people think that that's why alpha chromosome active domain can act as a glucosidase glucosidase. So even here you can see before our paper published, there's a lot of high level journal journal that they published alpha chromosome have beta glucuronidase activity or sialidase activity, right? So the people all think that alpha chromosome can the soluble alpha chromosome is a glycosidase, and they can act independent to the FGF23. So, but we take a very carefully look into the structure, we can find that if you compare the structure with closely related protein, the only protein that from closely family members with proven the glycosylamidase activity. So they have 2 different glutamic acids, which is the catalytic essential for the activity, for their enzyme activity. So no matter KL1 domain or KL2 domain, they only keep 1 glutamic acid here. For the KL1 domain, you can see the E165 is mutated to the N239. And for the KL2 domain, the E373 is mutated to the serine 872. So they only keep 1 glutamic acid. This is the first. And then we just did a docking model to dock the KL1 domain and KL2 domain to the glucose. We can find that for the KL1 domain, there is no pocket for glucose to bind. You can see here. And this is a KLRP, which is closely related protein. So you can see their pocket is very suitable for glucose to bind to there, and they can hydrophobic it. So, and for the KL2 domain, this binding site is very, very open and large. So the glucose cannot bind to here very stable, and they will directly, when they bind, they will directly go outside. So it means alpha-chlorosil is a non-enzymatic molecular scaffold in here. And we also use a substrate for those that published activity, which beta-glucuronidase or sialidase. We use their specific substrate, and we can find that the alpha-chlorosil is no activity for the glucuronidase activity in there. So we think that that's why we explain that why when you knock out alpha-chlorosil or FGF23, the phenotype is totally the same, because all the alpha-chlorosil activity is dependent to the FGF23. So then we go take a very close look into the binding interface of FGF23, Fgf receptor 1, and alpha-chlorosil. The red one is Fgf receptor FGF23 bind to the alpha-chlorosil. You can see here it's this most is the C terminal of the FGF23. Another rest is the red is the core domain of FGF23. So you can see here FGF23 it forms a lot of hydrogen bond with the alpha-chlorosil. Another part, this pink part, is I told that alpha-chlorosil, their receptor binding arm binding to the receptor. You can see here is a large area for them those two guys to bind to each other. So this like if you take imagine, you can see that those three proteins, they like three good friends. Chlorosil use their hands to to hang the FGF receptor, and the receptors FGF23 uses his hands to hang the alpha-chlorosil, and then these three get together, right? So this is like a family here. And one thing is that we find that FGF23 is only can active the FGF receptor CS form. So that's why it's like this, because there is a very important amino acid here, which is serine here, which can form a two hydrogen bond with FGF23. And you can see if we do the sequence alignment, you can see all the CS form of FGF receptor is serine here, but for the BS form, it becomes a big protein, a big amino acid is called tyrosine, right? So when we mutant the FGF receptor 1C, the serine to the tyrosine, you can see if the Y-type FGF receptor 1C, we can stimulate with FGF23. But if you mutate this serine to the tyrosine, their signal is totally lost. FGF23 cannot bind to the receptor anymore, even they have alpha-chlorosil. For the last, we just to measure the binding interface between the alpha-chlorosil and the FGF23 here with the C terminal. So we just to check the important amino acid which can forms a important interaction between those two molecules, and we mutant those important amino acid, and we can see it lose the activity of also this we mutant alpha-chlorosil, this we mutant FGF23. Both of them can lose activity. It means our structure is true. And for the FGF receptor 1C, it's more easier because their interaction is very big. So we directly cut this receptor binding arm immediately. So when there's receptor binding arm when there is a alpha colossal active domain together with Fgf receptor 1c alpha active domain, you can see they forms a dimer here in the red. But when we cut the Fgf receptor binding arm, you can see the black here. They cannot form a dimer anymore. So it means when we cut the receptor binding arm for the alpha colossal, they totally lose the Fgf receptor binding affinity. And you can see if we cut the receptor binding arm, no matter with actual domain alpha colossal or transmembrane alpha colossal, both of them can lose the activity. And we also generate that alpha colossal with actual domains that dirt the receptor binding arm, which means they cut this truncated this receptor binding arm, they can still act as a FGF 23 ligand check, which means it can use as a FGF 23 inhibitor. How? Because even we truncated this receptor binding arm, even this alpha colossal dirt receptor binding arm cannot bind to the FGF receptor anymore, but it still can bind to the FGF 23, right? So when we inject it into the mice, the active domain, the delta receptor binding arm truncated alpha colossal bind to the FGF. So the FGF cannot bind to the Y-type alpha colossal anymore. So it means they can inhibit FGF23 signal. You can see here, if we add the alpha colossal truncated receptor binding arm with a gradient of concentration, the signal goes down and down and down and down. So it means this alpha colossal that we can use it, this kind of alpha colossal, we can use it as a FGF23 inhibitor. And in the last part, we did another research is to what? I don't know everybody here, do you still remember the heparin or not? So at the beginning, we think that heparin is FGF23, or maybe not even FGF23, or the whole FGF, endocrine FGFs. We think that they have very, very low FGF heparin binding affinity. So we think heparin may not works there, but when we take a very careful look here, even FGF23 have a very, very low heparin binding affinity, but it still have a measurable binding affinity for heparin, especially FGF23. So we think that maybe heparin is still doing something in there. So we did a SEC mouse study, which we measure the molecular weight for the complex. When we measured first, we didn't add heparin. We just added this ternary complex, FGF23, FGF receptor, alpha colossal together. We found that the mass weight is only 150, around 150 kilodaltons, which is totally 1 to 1 to 1. They didn't form a dimer. Because before I told that FGF receptor, they want to signal transduction, they must form a dimer. So we think maybe heparin is doing something there. So we add like a gradient with concentration, we add the heparin like one ternary complex with 0.25, 0.5, 1 to 1, and 1 to 2. We found that when the heparin comes, they forms a dimer. The mass weight goes to 320, and you can see the signal. If we with the heparin, we add the heparin more and more, the signal goes higher and higher, and even we 1 to 1, it goes to highest, and even we go to 1 to 2, nothing changes. So it means it can form a 2 dimers when we add the heparin. So it means heparin is working. And we also mutant the FGF23 heparin binding site and the FGF receptor 1C heparin binding site. We find that if we mutant those binding sites, the heparin cannot make this dimer anymore. So now we know the heparin is doing something there. So why before we didn't find this question? Because before we didn't add heparin. When we do the cell study, we didn't add heparin buyout. So because of before we use a long a not correct cell line. Before we use HEC293 cell lines, but HEC293 cells can express heparin by themselves. So we change another cell line, which is called BUF3 cells. This BUF3 cell line is FGF receptor alpha colossal and heparin sulfate triple deficient cell lines. Then we can see if we didn't add heparin, even we add FGF23 alpha colossal actual domain, FGF receptor, or alpha colossal transmembrane domain, there's no signals. When we add heparin, the signal comes. So it means the FGF23, even they need alpha colossal to bind to the receptor, it still need heparin to form a dimer so that signal can come. So then we mutant this receptor, the heparin binding site of receptor and FGF23. We can see that they lose the activity. If you mutant this, they lose the activity even you add the heparin. So then we get a dimer, we dimer model. This model is we predicted by the computer is a predict. So you can see that in the part of this dimer, this is our ternary complex structure. The alpha colossal, they bind to the receptor and bind to the FGF23 and the heparin. So the totally is like the FGF, paragram FGF. The alpha colossal is like a co-receptor. They help FGF23 to bind to the receptor. And then the heparin helps those two ternary complex to form a dimer. So this is the dimer model we get. And actually our work is finished, and we submit this paper to the Nature, and one of the reviewer came. I think, do you know who is him? He is the Brandel. You remember that another model of FGF receptor, paragram FGF receptor, which said their heparin is in the middle of two ligand. So he think why you didn't try that dimer model and see maybe it's act like that or that dimer model. So according to that reviewers question, we did another predict another model, which is 2-2-2-2-2-1. So one heparin in the middle and two ternary complex in the left. And we found that this pink area is that when we do the model, use our model, we can see these two receptors, they will have interact with themselves. So here we can see if use the model by the Brandels, this pink arrow cannot useful anymore. So we did this kind of models, and we mutant those important amino acid to alanine, and then we found that when we mutant those secondary receptor binding site, they lose the activity. It means those two pink, the pink arrow of the receptor, they did a very important role in the interaction in the dimer. So it means their model is actually not correct. So we put those results into our revised paper and sent back to the Nature, and fortunately Nature take it. So totally we get the FGF23 models where we know how FGF23 is bind to the receptor and active the receptor. This is the whole work for the FGF endocrine FGF. Actually, we also put this structure into FGF19 and into FGF21 to check whether they are working as a same model or not. And fortunately, they work totally the same. We mutant those important interaction amino acid for those two endocrine FGF, and we found the same result. So at last, there's a summary is that first, the soluble chromosome is act as a co-receptor for FGF23, and alpha chromosome is a non-enzyme molecular scaffold, and alpha chromosome can interact with FGF receptor by its D3 domain and FGF23 by its C-terminal tails. And at last, FGF23 signal is both alpha chromosome and the heparin binding heparin sulfate dependent. So here I will introduce a guidance for you. This is a guidance that for the from the Greek. So he is doing the job to to serve the lifespan from the babies to the old. So the alpha chromosome is act like the same. They use the FGF23 and the receptor to make the longer of our people's lifespan. So that's all. Thank you very much. Okay. Hello? Oh, so it's finished?
Okay, doctor, thank you.
Okay, thank you. Bye-bye.
Thank you, sir. Goodbye.
Bye-bye.
Goodbye.